Well, hello, everybody. I'm Laura Catena. Buenos días to our friends in Argentina. Bon après-midi in France. Good evening to our English-speaking friends. Okay. So, well, first of all, welcome everybody to this wonderful seminar today about the science behind terroir. I am so excited about the group of speakers and about all of you joining us today in this journey to study terroir. First of all, I want to tell you what is the inspiration for this session. When I started working with my father 25 years ago, I was selling Argentine wine, and many people around the world had not heard of Argentine wine. They didn't know we had been producing wine since the 16th century. And I encountered many people who told me that all Malbec was the same, that all the wine from Mendoza was the same, that there was only terroir in places like France and maybe only in Burgundy, where you have those beautiful climats and crus. But I knew that we had terroir in Mendoza because I knew that if I tasted the grapes from a vineyard in a certain altitude, another altitude, different soils, even within the same parcel, I knew they had different flavors. So I've been on this journey for 25 years through the Catena Institute of Wine, which I founded in 1995 to try to understand the terroir of Malbec. But the reason we're here today is not only to celebrate the terroir of Malbec, which is a very exciting thing. As you know, we've published research, a very extensive research of the terroir of Malbec in Argentina and shown the differences of terroir, but also here to start a movement to understand the terroirs from all over the world and to encourage collaboration among different producers, small and big, and research institutions, both local and international. Because I firmly believe that for there to be a wine future, a future as beautiful as what we have today, where there are so many different wines from so many different places, and so many people live from wine and enjoy drinking wine, with climate change, we will have to employ science and research in order to continue to drink these beautiful wines and to understand the winemaking in each region and how we will confront these issues ahead. So for the first session, we have some great panelists to talk about the climate effect on terroir. We have Fernando Buscema from the Catena Institute of Wine, Dr. Benjamin Bois from the University of Burgundy, and Dr. Gregory Jones from Linfield University. Welcome, Fernando.
Thank you, Laura, very much. In the 2000s, different experts told us that we had no terroir in Mendoza, that all Malbecs were equal. Although the experts had a lot of information from other regions of the world, the Catena family had been growing vines in Mendoza for almost a century. And our experience indicated that there were unique wines that had unique characteristics associated with a particular place, wines with a sense of place. Laura proposed me at the Catena Institute a great challenge to find an objective way to measure the terroir and thus be able to confirm or not our feelings. I went to UC Davis to do my graduate studies and work with Dr. Roger Bolton on the most extensive terroir study that has been published to date, 42 plots of Malbec between California and Mendoza, plus a dozen in Washington state. Let's look at this graph for a minute. There are two lines that intersect in the center and divide the screen into 4 quadrants. On the right, we see some gray signs that refer to geographical indications of Mendoza, like San Carlos or Tupungato, while on the left, there are black signs that mention AVAs of California, like Napa or Sonoma. There are many small dots that indicate chemical compounds, sensory descriptors, and climatic characteristics that are present in some places, but not in others. The first conclusion we reached is that it is possible to find Malbec wines with unique characteristics, both chemical and sensorial, in Mendoza and California. Mendoza wines also had a greater number of associated sensory descriptors, suggesting that they might present a greater complexity than those of California. Floral notes, especially violet and red fruit, distinguish Mendoza geographic indications, while aromas of black pepper and dry fruit predominated in California IVAs. We were even able to distinguish the wines of different geographic indications in Mendoza. If you give me a bottle of Malbec without oak aging, without any other additional information, it's very likely that I can tell you where the wine comes from. There is a series of papers that we published at the Catena Institute about this study, with the intention that other producers in Argentina could thus recognize the regions. To our surprise, this methodology also became a reference that researchers from the US, Chile, and Australia used in Pinot Noir, Cabernet Sauvignon, and Shiraz. One question we ask ourselves with Nicolás Catena, Laura's father: how many years does these chemical and sensory fingerprints of Malbec wines last? After 5 years of aging, we were able to confirm that it's still possible to distinguish Malbec wines from different regions in Mendoza. Currently, we're publishing the results after 7 and 8 years of aging, and I promise that as soon as they're available, we will send you a copy. Very proud, I showed Laura all these results that we have obtained at the Catena Institute, and after thinking about it for a while, she told me, Fernando, you know, so that we can really conclude that we can measure the terroir, we need to know what happens in vintages with different climatic characteristics, even in vintages in which we do not need to irrigate at all. This led us to establish new collaborations with Dr. Bottini from CONICET in Argentina and Dr. Gregory Jones from Oregon. And I'm going to let Greg tell you more about the challenges of this project and some of its results. Thank you very much.
Thank you, Fernando, and welcome, Greg.
Good morning, good afternoon, good evening, no matter where you are in the world. It's a pleasure to be with you today. First, I'd like to say how much of a pleasure it was to work with the Catena Institute and colleagues Roy, Fernando, Ruben, and Ariel to collaborate on this research. And also a pleasure to carry out research that grew out of the vision of Laura Catena's idea to better understand the sense of place, or terroir. As a climatologist, I have been examining the role climate has in growing grapes and making wine for over 25 years. During all of this work, I have found that attempting to understand the uniqueness that a site brings to wine has been challenging. But the work like the one we are discussing today and others have started to unravel some of this mystique. Our study that we're speaking about today benefited tremendously from examining one variety, Malbec. It also benefited from the fact that we were looking at this situation in a region, Mendoza, where site characteristics were dominated by changes in elevation over very short distances. And this strongly influences the climate signal. But to do the work that we carried out, we needed spatially appropriate quality climate data, which is not always easy to come by. Combining data from the Catena Institute of Wine, the Mendoza Department of Agriculture and Climate, and TerraClimate, a spatially integrated global climate data set, we were able to categorize the weather climate signal for these 23 parcellas shown on the map on the left-hand side of this figure. An example of the TerraClimate data that we worked with is shown on the map on the right side of the figure. And the data are for growing degree days, a measure of heat accumulation and ripening potential. And the data show that ripening categories from quite cool at higher elevations to very warm at lower elevations can be seen in parcellas that are separated by maybe less than 80 kilometers. An additional benefit was to be able to carry out the study across 3 vintages, in which Mendoza experienced one with significantly different conditions from a weather and climate perspective. The 2017 and 18 vintages showed climatic characteristics similar to the historical average of Mendoza, while the 2016 vintage experienced 2.5 times the normal rainfall and had very humid conditions throughout the growing season, which are not common in Mendoza. These differences in vintages were seen in the composition data, but also showed consistently across the parcellas, meaning that their terroir signals were consistent in spite of very different climate conditions. During the rest of the session today, we will be sharing more about this study and other research and how it has increased our understanding of terroir. Thank you.
Thank you very much, Greg. Benjamin, will you tell us about your amazing septentrion study?
All right, thank you, Laura. Hi, everyone. I'm thrilled to see there are so many people following the webinar at the moment, and I'm very happy to share with you my experience within our Septentrion project, which is a worldwide project that was made possible with the collaboration of the Catena Institute of Wine, and also the help of many grape growers worldwide. We're not talking about we've been talking about what's going on in Argentina, and then Greg, who's from Oregon, talked about a study, a very interesting study at Terroir at a local scale in Mendoza and Valle de Uco, and now we're but in the place where you live, Greg, there is also a lot of Pinot Noir which is cultivated, and you can see the background. I'm in Burgundy at the moment, and in Burgundy we cultivate a lot of Pinot Noir. We have some common characteristics, if I can say so, with Argentina, because there is a leader cultivar which is Pinot Noir, and of course Chardonnay for white. And the cool thing with those two cultivars, they are cultivated in many regions worldwide. So what we wanted to see with the Septentrion project is a little bit similar to what Fernando has been talking about in comparing how a single cultivar can behave in different environments. And for this, we're collecting samples from different regions all over the world, from France, of course, for Burgundy, of course, but also Champagne and southern region of France, even in Bordeaux and Gaillac. There is a little bit of Pinot Noir in Bordeaux. Thanks, Kees, for helping with that. And but also in Portugal, Italy, Australia, California, and of course Argentina, where, as Greg said, we have a wide range of elevation and obviously a large diversity of terroir condition there. So basically, we collected samples. Very simple. We're collecting samples of juice from 80 plots in different vintages, and then we analyzed them with a mass spectrometry, which is a very big device that helps us having a lot of information from those samples, more than 20,000 molecules that are identified. Part of them that we don't even know which kind of molecule they are and what role they play in the biology of grape and in the taste of the wine. By comparing this, we can identify the effect on the environment, and I work a lot with a climate and viticulture relationship, and what we want to see if there are some insights about the effect of climate in different type of climate condition, dry climate condition, warm climate condition, humid climate condition worldwide, by comparing samples. Even from Germany, I forgot to say so. And what we see there is a two-scale kind of results. We see that there are, of course, large difference between countries, between wine growing region between those countries, but also within a same wine producing region. For instance, Burgundy, we clearly see the different sub-region, what we call climat, the terroir in Burgundy. We call that climat in Burgundy. We can see the difference with those kind of analysis, metabolomic analysis. So we started this collaboration, and I'm very, the cool thing with working with the Catena Institute of Wine is that it helps us, of course, having a large diversity. We talk about terroir in Argentina, access to different Pinot Noir and Chardonnay from different locations. But also there are two important points that I would like to stress. First, the team, the group, Fernando, Laura, Roy, and so on, share a real interest in science and research, which help us exchanging and our results and talk about our results. And second, you also have a great technical support, and having such making such a program with, you know, analyzing and collecting samples from all over the world wouldn't have been made possible without this collaboration, because we also try to see how the fingerprints of climate and environment translates from the juice to the wine through microvinification. And so we handle microvinification in Argentina, in Champagne, and also in Champagne this year, and also in Burgundy. So more to come in the next in the next month. And again, thank you for the collaboration, and thank you for your attention.
Well, thank you, Benjamin. This is such an ambitious project that you've undertaken, and I can't wait to see the results, because it will be so exciting to see, you know, the Pinot Noir variety and Chardonnay from all over the world. So I have one question, and then we will move on to the next session. This is a question for Greg. So the question is, should altitude become part of the definition of terroir along with temperature and soil?
Thank you very much for the question. Of course, I agree. But if you're categorizing climate correctly, climate will capture the effect of elevation. But it is really good, I think, to include the idea that elevation plays a role. It's just that if we measure climate correctly, we're measuring elevation at the same time.
Thank you. That clarifies things for me, because I also, this was a question in the panel, but I've also asked myself that question. But you're right. If the altitude is already included in the climate, then maybe you just state the altitude. The one question I have about this is in terms of sunlight intensity.
Yes.
That might be different with altitude, and maybe the question should be: should sunlight intensity also be included in the definition of terroir?
Yes, Laura, I agree. I think sunlight intensity, ultraviolet radiation, and how that affects ripening and different style characteristics, I think is very important to include.
Very good. Well, thanks. This was an incredible panel, and we will see you again later at the end for the Q&A. We will now move to the panel on the soil effect on wine. We have some amazing speakers: Roy Urvieta, our Director of Winemaking at the Catena Institute, Dr. Kees van Leeuwen, who is the Head of Viticulture at Bordeaux Sciences Agro, and Dr. Patricia Piccoli, who is a professor at the local University of Mendoza and a very well-known and respected researcher in soil microbiome in Mendoza, Argentina. So welcome, all of you, and Roy, please start sharing your incredible information.
Thanks, Laura. Well, I'm going to show you some chart of the study that we published in Scientific Reports. This is a heat map. It's about the concentration of phenolic compounds, no temperature or weather data. In the horizontal axis, we are going to find different phenolic compounds that we measure. These compounds are responsible of the color in red wines, the mouthfeel sensation, the aging potential. They are very, very important for the quality in the red wines. And in the vertical axis, we have the geographical indication of Mendoza: Rivadavia, El Mirador, La Libertad, from the east of Mendoza, and Gualtairi. The color in this chart indicates the concentration of the phenolic compounds. The red is high concentration and blue is low concentration. It's very easy to find see the phenolic profile. For example, if we compare Rivadavia and Gualtairi, Rivadavia is the first one and Gualtairi is the fourth. Rivadavia has low concentration of delphinine-free glucoside, is the one anthocyane responsible of the color, and Gualteri has a high concentration of that anthocyane. But in general, the phenolic profile are so different. Rivadavia has some blues and red, and Gualteri has other blues and red. That we call the chemical fingerprint. Each region has a fingerprint that we can measure, and it is different. These regions sometimes are close to each other. If you get your car and drive from Rivadavia, the east of Mendoza, to Gualteri, it's a little bit more than 1 hour. You go to the Winkler 5, the warm climate, to the Winkler 1 or 2, the cool climate. Winkler is a bioclimatic index to compare the climate for the region around the world. That is amazing because it's like going from Burgundy to Languedoc in 1 hour. In this chart is the parcella level. Here we have 2 axes. In this case, we measure 27 phenolic compounds, but it's very difficult to plot 27 different compounds together in one plot for the parcella level. But what is a parcella? It's a small plot or parcel less than 1 hectare in the vineyard, but the soil is homogeneous in that parcella. In this chart, each circle is a small parcel. For instance, the green circle are from Rivadavia, and the orange circle are from Gualteri, the similar example that we saw in the previous slide. Clearly, not all the parcella are the same from Rivadavia and Gualteri. Basically because in this chart, if 2 parcella don't overlap, we can tell that there is a statistical significant difference. Well, maybe you say some years is different, another year is not different, but in this plot, in our statistical analysis, we combine 3 years of study, 2016, 17, and 18. It's like the unique fingerprint is consistent in that parcella. In the vintage that we don't have to apply irrigation, we have to apply irrigation like 17 and 18, and some vintage like 16 that we don't have irrigate at all. This is extremely consistent in different years, in different climates, with irrigation and without irrigation. Even in the same vineyard, we observe there are different parcella with different phenolic profile, different fingerprint. In the particular vineyard, assume that the climate is similar, they have the same elevation, and the plant material is the same. But what is the main reason for that difference in the phenolic profile in that fingerprint? We think it's the soil. And some parcella, like Adriana vineyard, we have deep soil, and some other very close to the first one, around 50 meters. A part of the deep soil, we have parcella with shallow soil. We can see the stone in the surface, like in this picture. We have deep soil and shallow soil. But the wine that we obtain from this parcella is consistently different. In the vintage that we have to apply irrigation, and vintage that we don't have to apply irrigation. Well, the soil matters, and our friend Kees talk more about this topic, and Patricia later going to explain what is the role of the microbes in the soil, particularly in these parcellas in Adriana vineyard. Thank you.
Thank you very much, Roy and Kees. Please teach us about soil and wine.
Yeah. Thank you, Laura, and thank you for your invitation. Terroir can be considered as an ecosystem, and there are several compounds which are important in terroir. And one which is obviously important is the climate. Another important compound of the terroir is the soil. But terroir is not only about the natural environment, it's also very much about the vine. And so the vine should be placed in the center of this ecosystem. And that's why I like to refer to terroir as an ecosystem. In biology, an ecosystem is the interaction between a population of living organisms with the natural environment. And terroir is exactly that. Terroir is a population of vines interacting with the soil and with the climate. Now, the climate acts through a certain range of temperatures, a certain amount of radiation, available water, and CO2. The soil acts on this ecosystem through the availability of water in the soil and nutrients. And among those nutrients, nitrogen is of particular importance. Now, of course, terroir is not a natural ecosystem. It's a cultivated ecosystem. And that means that also the wine grower matters, because the wine grower makes a certain number of decisions in terms of the plant material he's going to use and the viticultural management techniques. And now, for instance, for the plant material, the variety will always be chosen very closely in link with the climatic conditions. And for instance, in a cool climate at higher elevation in the Andes, growers will pick early ripening varieties like Chardonnay or Pinot Noir. And in warmer locations, they will choose later ripening varieties. The rootstock is always carefully chosen in relation with the soil, depending on the lime content or the available water, for instance, and more generally speaking, the fertility of the soil. And then the grower also applies management techniques, like, for instance, the training system, including the density. For instance, the row spacing will be related to the amount of available water. If water is scarce, then the density will be lower, and if water is not so scarce, like it can be in Burgundy or in Champagne or in Bordeaux, then the vines are planted closer together. Canopy management can be related to the amount of incoming sunlight. Floor management, depending on the amount of available nutrients. For instance, cover crop can compete with the vines for nitrogen. And water management is, of course, also very important. Like Roy said, in a dry vintage like 2017 at Catena, there was no irrigation applied. But in very dry vintages with as little as 100, 150 or 200 or 250 millimeters, a little bit of supplementary irrigation is needed. And of course, the nutrients can also be adjusted by fertilization. So in this ecosystem, all the interactions are very, very important. And the terroir effect actually plays through the interactions between the plant material, the climate, the soil, and the plant material and the management techniques. And these interactions will yield a certain grape composition. And then after vinification and aging, this will yield a certain typicity and quality for the resulting wine. So if there is any effect from the soil and the climate to the quality and the typicity of the wine, it is necessarily mediated through the vine. So it's very important that in terroir research to put the vine in the middle of this ecosystem. Now, if we want to measure the terroir effect, then we need to break down the effect of the climate and the soils in measurable variables. So if we do terroir research, we are going to measure a range of temperatures. We need to know the water status of the vine in relation to the soil and the climate. And there are different techniques like water potentials to measure the water status of the vine. Incoming radiation can be measured, and also vine nutrient status, and in particular nitrogen status, can very precisely be measured. So once we have identified the important factors that act on the terroir expression, and we know that we are able to measure them, then we can understand how the soil and the climate, how the terroir influences the taste of the wine. And typically in the example of the two soils that Roy just previously mentioned, in a shallow soil, which is a poor soil, nutrients are less available, there's less water available, and the grapes will be smaller, the berries will be smaller, and the grapes will be more concentrated, and the wine will show more concentration. But in a deeper soil, where the berries are a little bit bigger, in a relatively warm climate, the vines can show very nice finesse and freshness on the palate. So the type of the wine produced will be very, very different according to the soil type. And this can all be explained. There's nothing mythical about terroir. Terroir is just very much about basic agronomics. There are resources in the natural environment, temperature, water, light, nitrogen, and the vine is going to use these resources. And depending on the place, these resources are more or less available. And then the vine grower plays with the plant material and the management techniques to optimize the output in terms of quality and typicity.
You make it look so easy, Kees.
Thank you.
So yes, so many variables. It's actually, you know, as a physician that looks at medical research, you know, I think actually that viticultural research is so complicated because you have so many variables and it's so hard to control for all the different variables. And that's why I find the research, you know, that Roy and Fernando did at the Institute, and, you know, the work that you've done, Kees, and I've read so many of your papers that are just, you know, a master, you know, of trying to control for these different things so that you can reach conclusions. But it's very complex. And I really love the word ecosystem to describe terroir. I think that's wonderful. I will start using it, if you don't mind, from now on. So, well, I would like to now introduce Patricia Piccoli, who is a microbiologist. And actually, Patricia, we already have some questions from the audience about the impact of the microbes on terroir. So I think they will be very interested in your presentation.
Okay, hello. Thanks, Laura, to inviting me to participate in this meeting. It is a pleasure to be here sharing this space with all of you. Plants, I want to notice that plants, like human beings, are holograms, that is, not individuals, but ecosystems. In animals, a plethora of microorganisms is present in guts, while in higher plants, there are mainly located at root levels and rhizosphere, and also in intercellular spaces of the root. If terroir, I agree with Urvieta and Leeuwen, as if terroir is determined by the interaction among soil, climate, genome, and winemaking practices, then genome is hologenome, in my case, metagenome. Okay, there are two approaches, metagenomics profile and microorganism target search. In grapevine, we started to use the second one by searching conspicuous plant growth promoting rhizobacteria, henceforth PGPR, in a specific parcel at Adriana's vineyard, from root and rhizosphere at different soil depth of Malbec vines. Okay, let's see a summary of the results. By screening the PGPR like strains, 11 different species were identified and sequenced. From them, Bacillus licheniformis and Pseudomonas fluorescens were selected on basis of their characteristics as PGPR. Okay, we mimicked a great plant ecological system by cultivating in vitro Malbec virus-free plant and inoculated or not with bacteria. Well, as you can see there, the picture shows on the center right that is important to know. Right here, 45 days, all BTs, vinifera Malbec plants, bacterized here with Bacillus and all Pseudomonas, demonstrating the ability to colonize grape roots and stimulate overall plant growth. Figure at top left shows you shoot length for plants bacterized or not, and you can see differences. Both bacteria, this is important to me because are able to produce abscisic acid, AA, and also gibberellins. Okay, here is another, I kind to remember that this is different that I did the last, okay? So here also there is gibberellins, A1 and A2 and 3. Okay, AA, I want to notice that, as say there, augmented root length, and ABA, water loss rate, stomatal conductance, that is reflected in these pictures, where I want to say, for example, that the level, and it's related with that, the level of ABA and in Pseudomonas was approximately 76-fold more than the control, and Bacillus 40-fold times. Okay, so we can say that this elicitation of ABA levels diminish plant water losses, as you can see in the left, in the right, excuse me. And I want to see that if you see into these arrows, you are able to see that the time period in which plants inoculated with both bacillus lost approximately between 4 and 10% less water than controls. So also monoterpenes and sesquiterpenes are species mice, augmented bacteria plants, so increasing compounds related, that is important, with flavor, as show Urvieta and shows also Fernando, augmented the bacterial plants, increasing flavor and also photoprotected function by augmenting ROS deactivation, as you can see here in the right tab. So I want to add that also terpenes, mono and sesquiterpenes, are able to for inhibition of the growth of some pathogenic effect of, for example, Botrytis cinerea. As you can see here, the difference between control, this leaf with cinerea, and of course this apply with or bacterize with bacteria, both Pseudomonas or Bacillus. Okay, thanks a lot, and it was a pleasure, of course, to be here sharing with you this meet.
Thank you, Patricia. I of course, I'm obsessed with this research. As a doctor, I love to hear about microbes, and I think that just like what you showed about these PGPR, plant growth promoting rhizobacteria, which rhizobacteria means that they live with the roots. Look at what they do for these plants. I mean, look at the plant on the right. It's got more green, it's got more terpenes, it's got this abscisic acid, which is a natural product of the vine that helps it withstand water stress. I mean, I think that we are going to learn so much more in the future about how microbes, bacteria, fungi are so important for agriculture. And, you know, what I find very exciting is that we're finding similar things for people. You know, medicine is studying the effect of microbes, and most people don't know this, but we humans have more microbial DNA than human DNA, because we wouldn't survive without these microbes, and the vines wouldn't either. And I think that for sure, and there was a question in the chat about should the microbiome the microbes in the soil and on the vines be considered part of the terroir. And I think they do need to be part of the terroir definition because they are so important. But like you said, we still don't know enough about them. And hopefully we will learn more in the future through research like what you're doing, which is so fascinating.
Okay, so, thanks a lot, Laura.
Thank you, Patricia. Thank you, Roy and Kees. So now we will go to our last session right before the Q&A. And I'm going to invite Jane Anson to wrap it up for us. But first, I want to acknowledge some of the research institutions that we worked with. Roy, if you could show this slide, thank you very much. So I want to emphasize before we finish that we producers of wine in the world need to collaborate, local institutions, institutions from around the world. I have here some of the logos of all the institutions we've collaborated with, and I want to especially commend Kees van Leeuwen for his work with Yves International Viticulture and Enology Society, which the Catena Institute belongs to many wineries all over the world, in France, in Bordeaux, where Kees is working, belong to this group of wineries that are interested in collaborating in research for preserving our beautiful world of wine. Also, Yves publishes the Yves Technical Reviews, which are based on research done either by research institutions, universities, or by producers. And also Eno1, which is an open source journal similar to Scientific Reports, where we published the Malbec study. And I urge you all to get online, to read some of these papers because they are open source, and to get involved in this research that is going on in the world and that can help us preserve the flavor of wine. So Jane, can you put it all together?
Certainly, well, I'll do my best. Thank you so much for asking me to be a part of this afternoon. It has been so fascinating listening to those experts layering the information one on top of the other, and every presentation helped make the one before mean more. Often you read these experts in on their own with their own research, but seeing how it comes together is just very inspiring. So thank you to the Catena Institute for really putting all this together. Also, it's been interesting for me personally because much of the last 5 years of my professional life has been trying to put this together, to understand and to clarify what all of this means. And in fact, two of these panelists, Kees van Leeuwen and Benjamin Bois, were instrumental in the research for my most recent book, Inside Bordeaux, where I was trying to clarify the link between terroir and why the wines of Bordeaux taste the way they do in the glass. So it really just so fascinating for me hearing all of this. And I'd just like to emphasize to everybody listening, if you haven't yet gone to read the original study by the Catena Institute, I thoroughly recommend you do so, because there are so many threads in there which you can pull on to learn not just about the link about soils and typicity, but as we've been saying, altitude, vintage, winemaking. And you just learn so much simply by breaking down the methodology that they have used for this research. Now, every line was giving me more inspiration for things that I wanted to continue with, so I really recommend it. So my role this afternoon, in a tiny way compared to all these brilliant experts, is just to try to make sense of this as a non-scientist, and to try and kind of help with all of us to understand what's the interest of this for wine lovers. And I point to a headline from Wine Business magazine that I read just last week, an article by Lance Cutler, which was called The Provable Theory of Terroir. And that seemed to me to sum up really at the heart of what Laura and the team here are trying to do. The idea that by proving the link between sight and taste, it will help consumers seek out more meaningful wines. It will help the winemakers deliver more meaningful wines, but it will also help draw attention to high quality sites that potentially over the next decades, we're going to need more and more proof of why they need saving, supporting, and paying for in many ways. Now, I know from experience as a wine critic that it's much easier explaining wines to other people if you can show that there are tangible reasons why they taste the way they do. And it's why one of the reasons that I loved Kees saying quite clearly, it's not mythology. You know, there are specific reasons why things taste the way they do. And I would like to add that the fact that this study comes from Argentina is massively helpful, and America is massively helpful in lending credence to the results. Because there is a tendency, or there could be a tendency, if this had come from France, proclaiming the preeminence of French soils, that it could have been dismissed. So again, really, Laura, many congratulations. Anyway, I'm going to look a little bit about the relevance of this study for the future of wine regions. And I would like to point to a basic economic fact, which is market demand. If there's no fine wine market consumers who want it, then it's very, very hard to kind of support these wines going forward. And if this research helps underline the link in the mind of consumers between terroir and high quality wines, then it will help to sustain the demand for these wines. And this has been true for generations in France, but often it's been dismissed as marketing or as, you know, having the results of having powerful people with money at the right time in the right places. But the more that we see things like this, the more you realize why great wines are where they are. And I just want to point to a tiny little study which I looked at inside Bordeaux, which I know Kees doesn't always necessarily agree with, but I'm going to talk about it anyway. The 1855 estates in Bordeaux, they're probably the best example of wines which we have said over and over again that they are so successful because they had wealthy, powerful people at the right time owning them. In the book, I looked at some research that was done by Pierre Becheler, who is a geologist, and he has identified 6 different terraces within the Medoc that are numbered from T1 to T6. And the really fascinating thing that I found, you find those all over Margaux and all the different appellations, but every single 1855 classified estate is on either T3 or T4. And that they were named that back in the 19th century when nobody had any idea about any of this. So these little tiny snippets of information, they're all part of the jigsaw of what makes terroir important, what makes special estates so special. And I would say that the importance of recognizing these viticultural sites worthwhile is worldwide is becoming ever more important. We're all aware of climate change, the risk for viticulture, and if we can make these, this real provable link between specific sites and the success of the wine that comes from them, then it's great for all of us. It creates an economic incentive to preserve them, and it helps all of us who love fine wine. So I would just finally like to add that there is another, I would say, huge education job to be done, which was absolutely talked about in this last panel, and it's one of the reasons that I really, really appreciated Patricia's talk, which is we need to make a link between living soils and terroir. So this idea, we're seeing a revolution that chemical fertilizers, pesticides and herbicides are the enemy of healthy soils, but we need to keep making that link. And the work that these guys are doing, I think really helps us. It seems logical to me that to have a fingerprint, then you need to have a soil which is alive and which allows that connection between the soil and the vine and the terroir to be made. So it's another big topic, but what I've loved about listening this afternoon is this is all about collaboration. It's all about one idea leading to another. So I look forward to following it, you guys, and your brilliance over the next couple of years. So thank you very much.
Well, thank you so much, Jane. You say so much in such a short time. Amazing. So let's start with some of the questions, and I will go one by one for the speakers. Question for Kees. Is it possible to talk about terroir without considering the use of indigenous yeasts?
Definitely it is possible, because soil and terroir, as I explained, through the effect of water, nitrogen, light and temperature, will modify the grape composition. So with grapes with a different composition, different tannins, as Roy showed, and different aromas, you will make a different wine, even if you use industrial yeast. There's nothing bad with using indigenous yeast, and maybe that can possibly add another layer, and maybe there are specific yeast in specific places, but even without using indigenous yeast, you can definitely make terroir wines, no doubt about it.
Thank you, Kees. Great answer. Question for Greg. If we take temperature into account, does this mean that with climate change and specifically global warming, the terroir designations will change? And the second part of the question, how much do the measurements need to change for us to consider the terroir to have changed?
Well, I think the answer really is as Kees has said, we are operating in ecosystems, and an ecosystem is a fluid, changing environment by which plants and animals adapt. And I think that, yes, as conditions change in the environment, then our vision of what that looks like has to change as well, because those ecosystems are changing. So we have to be able to be adaptable also in our viewpoints beyond this as well.
Thank you, Greg. Great answer as well. So question for Fernando. Do your studies also segregate data by Malbec clone? It seems different clones could be another important variable.
Thank you, Laura. That's a great question. In fact, we hold at the Catena Institute the largest Malbec collection in the world, and we are very happy to protect that piece of nature as part of our program. So it's really important to assess the impact of clones, and we've done that in a separate study, and they do play a very important role. And in the context of climate change, maybe some clones or a subset of this collection performs better in a particularly highly elevated vineyard, and in a different location with in a different ecosystem, a different subset of these clones might perform better. So preserving this collection is critical, so that in the future we can keep making distinctive wines, even though some climatic conditions change.
Thank you, Fernando. And just to, on the finer point of the question, we did not control for clones. So in the study, the Malbec study that we're talking about today, we were taking Malbec plantings. Some of them were Massal selections, some of them were clones. So that would be another study, which would be, you know, a very specific either population or clone in different places. So we do that next, Fernando. So now I have a question for, let's see, where's my question for Benjamin? Okay, while I find my question for Benjamin, I'm going to ask Patricia a question. What effect do herbicides or cultivation have on the rhizosphere micropopulations? You are on mute, Patricia. Unmute yourself.
Excuse me. Please, okay. Benjamin, if you are able, please, I would like to share with you this response.
Oh, concerning the microbiome?
Mm-hmm.
The role of the well,
I would like to see or to listen to your point of view, because you are French, and it's quite different relationship of soil in your country and in our country, because as we are young country, so we have not so many problems with pesticides, herbicides, and we try to protect so much because we are learning about the Asian countries like your country. For us, we try to minimize the pesticides or herbicides in the soil, because we know that this changes the native or the population, microbial populations, and that could be very undesirable for the plant, and also to have good, because I know that bacteria is just a little part, but it's not bacteria, it's the microbiome that is altering all the time the equilibrium if you are not taking care of it. So please try to don't use so much herbicides, try to don't use so much pesticide. We have natural, wonderful microflora that are able to repair also this kind of things. I would like to add that, for example, Johanna Dobereiner, a wonderful microbiologist from Brazil, she was the master of this subject, and she always tells us, we are younger countries, please ask to the producer, tell to the producer, tell to all the people that work in farms that soil is important, like climate. All is important. Don't minimize the soil and don't minimize the bacteria, the fungus, all the microorganisms in soil. Excuse me, Laura, I put.
I know, I love it. I think
I'm Italian.
No, I think that many people don't know that you and Benjamin know each other very well because you've worked with the OIV together. So Benjamin,
Yeah.
Tell us a little bit about your view of soil and
Right.
the microbiome and
Right. I think the first thing we have to say concerning the microbiome is like you have to consider that we are on the verge on discovering a whole universe, and with a microbiome. As you are, Laura, a doctor, you know that we're doing a tremendous a discovery with concerning what's going on in our guts. And in the soil, you got so much biodiversity that we really, at this moment, the microbiologists, excuse me, Patricia, I don't know if you share my opinion, but the microbiologists are just like, if they are discovering a new planet, and there are so many things. So there is to stress a little bit what's going on with microbiology and soil knowledge so far. I would say first that the International Organization of Wine and Wine, the OIV, is currently considering this very seriously, encouraging research on it, because you got to consider several things. First, you got a diversity, but within this diversity, what part of the ecosystem in the soil plays what role for the plant. So, and now we've got the capacity to analyze so many of those species of fungus and bacteria in the soil. We're at this very moment, we understand that you need an equilibrium in the soil. And to answer a little bit the desperate call of Patricia concerning the soil, what's going on in the soil and the use of pesticide and herbicide, I would stress that not only the use of chemical products are negative for the soil, but repeated use of plowing in the soil. And you know, where I live in Burgundy, we've switches in the 80s from excessive use of herbicide in the soil to plowing. But now we are on excessive plowing in the soil. It destroys also the life, the small worms in the soil are destroyed by plowing, and all the soil are compacted. So this is a big challenge now in Burgundy, and not only in Burgundy, in other regions. So you got, yeah, I agree with you. We have to learn about, but again, it's agronomy. It's doing things very sharply, and I'm pretty sure the viticulturists, the wine growers are aware of that and trying to find solution to balance and preserve the soil. I'm pretty sure the message is in all the minds now of grape growers at least in Burgundy, and I'm pretty sure in worldwide. But there's still a long way to go, and more specifically a long way in research to understand better the microbiome and its impact on grapevine biology and grape quality.
Well, thank you so much for that back and forth there. That was wonderful. So I have just one very quick last question, and then Jane, you and me, we have some questions for us, but we'll answer later. So Roy, could you explain a bit more what technique you are using to measure the level of phenolics? Last question.
Okay, well, in this case, in the last paper that we published, we use the HPLC DAD. This is the big machine that you add the samples, and then you can measure each phenolic compounds in the wine. For example, in the anthocyanins, we had 12 different anthocyanins, and we measured the concentration of each anthocyan. The technique is the HPLC.
Very good. Well, thank you, everybody. I promised we would end exactly on the hour. I can't tell you how much fun I've just had. I wish we could do this every day. And I can't wait to see you all in person soon, and keep on doing this incredible work. And all of you in the audience, we had over 400 people at some point. Talk to your friends, producers, and get involved in this movement of using science to preserve the nature and culture behind wine. So big kisses to all. Thank you very much. Adios, au revoir, goodbye. And
ciao, ciao.
I'll see you all soon with a glass of wine together.
Yes.
Bye-bye. Gracias.
Au revoir.
Au revoir. Au revoir.
Bye-bye.
Bye-bye. Bye-bye. Thank
Adios,
you.
adios.
Ciao.